All right, welcome everyone to the final presentations for the AI for Coral Reefs 2 challenge. We're very proud to present these results. We've worked hard for the last 10 weeks to come up with them. And yeah, to get started, I really like to thank Johan and especially from Reef Sport for organizing this challenge with us. So I hope you'll be as happy with the results as I am. For today, I'll do a quick challenge introduction. Then we'll hear from the first team who did the unsupervised approach. And then we'll hear from the other teams who will be talking about supervised approaches of coral reef monitoring. I've left some time in the end for some final words from Johan, and just as a thank you for everyone who joined this challenge. For the people who joined here today who do not know Fruit Punch AI yet, we are a global AI for good community, and we started this community because we believe that to solve humanity's greatest challenges, we need to educate AI engineers at scale with an ethical and sustainable mindset. We do this by organizing AI for good challenges that all are aiming towards the sustainable development goals. So we organize these challenges in wildlife, energy transition, or healthcare, for example. This challenge specifically links to the Life Below Water Sustainable Development Goal. And by organizing these challenges, we also educate AI engineers. For today, we're going to talk about the Coral Reefs Challenge, and the main problem that caused us to organize this challenge is that monitoring of coral reefs is very hard work and prone to errors. So coral reefs are one of the most important ecosystems on our planet, and almost 50% of the world population is dependent in some way on sea life. While sea life is mainly dependent on these coral reef ecosystems. Coral reefs are very large and hard to monitor. As one dive of 4 hours can sometimes result in 40 hours of manual labeling just to find out what the coverage of coral is on the ground and to decide on whether there is a growth in biomass or a loss. Also, corals dying has been a very large problem. So to bring this in, to map this and to give us some better information on how we should treat these corals, it is very important that we monitor it correctly. Gesundheit. And in order to do so, we're trying to use AI to improve the monitoring or at least the labeling of these coral reefs. As I said, the goal of this challenge is to automate the calculation of benthic coverage to monitor the biomass of coral reefs. So benthic coverage is basically all the life that occurs on the bottom of the sea. To solve this problem, we basically had two approaches. So we had the supervised learning teams who have used the masks and the labels. So using a given segmentation mask, they will train new models and try zero-shot learning approaches, like, for example, the segment anything model. For everyone who's not too familiar with machine learning, within machine learning, there's often two forms. Supervised learning is one where you have labels, and your model will learn from those labels and calculate losses based on the outcomes of the predictions. You can also approach these problems in an unsupervised manner, basically using no labels and letting the machine or letting the AI find patterns or clusters. So the unsupervised learning team is using visual clustering methods combined with point labels to create segmentations and look into augmentation techniques. We have tried multiple models, quite a lot actually, so I will let all of that explanation go to the teams. But there's been done a lot of experimentation, so if you have any questions throughout the presentations of the teams, please raise your hands and we will try to attend to those as soon as possible. We worked on this challenge for 10 weeks with 4 groups and 20 AI for good engineers with the combined effort of almost, over 1,600 hours of engineering work. So quite a big effort, and I'd say the results do show that. That said, I think we can get into the first presentation. So I will hand the screen over to Masum, I believe, for the unsupervised learning tools. Yes.
Okay, perfect. Um, all right. So thank you everyone for joining. I am Masum. Me along with Onea, Joanne here, um, and a few others I think should be on the call. We belong to the unsupervised learning team for the CI for Coral Reefs 2's challenge. And I'll just go ahead and tell you guys about what we did. Let me start with the data sets that we have been experimenting with and used to develop our solutions. Um, so for inputs, we have had images and point labels, which are manually annotated by the Reef support team and also other data sets annotated by other such organizations. And for um, we have also had dense masks, which we sort of use to validate our results, but to serve as inputs for training for the supervised learning team. They have been using images and dense masks. For the unsupervised learning team, we have been using the inputs as images and the point labels as inputs for training and inference. And to evaluate, we also use dense masks. I'm not sure in what capacity exactly the supervised team use these different data sets, but this is a general overview of what unsupervised learning team uses for the data sets. Um, this is this is an overview of all the different data sets we have been using, the number of images, number of point labels we have had, um, if there have been dense masks and the use cases. You will see for CView there were two different data sets. One has like 8,680 something images, which is something we haven't really used for building our solutions. We end up using the images, the 2707 number of images, out of which something about 1,054 images were excluded due to mismatch in labeling between CView and the Reef support teams. But regardless, we've still had a really good amount of data set to experiment with and build some really cool solutions, I think. Same goes with IBM and Revolution as well. We intended to use those for verification of generalization, um, but we haven't really felt the need to use those. So yes, let's dive into our approaches. I'll go about I'll start talking about the literature survey, and then we'll start diving into three main different approaches that we have experimented with, along with the roadmap, which are the label propagation, the SAM fine tuning, uh, or the zero shot segmenter, and then the interactive UI, interacting labeling UI, basically. Um, so from our literature survey, we found a few papers which really helped us in developing our solutions, and we were able to adapt their solutions, tweak it in some way, use it for our use case. A very important one was the label propagation paper, which is titled "The Point Label Aware Superpixels for Multi-Species Segmentation." You will hear me calling this particular paper PLASPIX, or the approach as PLASPIX, during this presentation. It's just very handy, and we found the name was pretty cool to say. But the approach basically, it's a very simple approach. It's, uses an algorithm to calculate superpixels, and also uses some feature extractor features to uh, basically segment an image. I'll go a little bit deep into it very soon. And then we have had the SAM papers, which is the segment anything model from Meta. We found specifically three different papers which were really helpful for us to develop the label propagator using SAM, as well as the zero shot segmenter using SAM. Um, the first one is EquaSAM. which we use to where we can fine tune separate decoders for each class, which we have done. There's AutoSam, where there we add a extra encoder to generate a rough mask, which then gets passed on um, with point labels, data images, or yeah, just point labels, and it creates a segmentation mask. And similarly, there is a different SAM paper or AutoSAM paper where they use similar approach for medical images, but they leverage a multi-mask setup, which allows segmentation with just a single model instead of using different models or different decoders for each class, which in our case would be the hard corals, soft corals, yeah, mainly two. So solution pathways, as I already said, mentioned, the label propagator. This is this is one of the approach where we wanted to use the point labels to generate dense masks for the image. Um, the idea is to generate additional data for supervised training process, or it can also be a handy utility for end users and interactive labeling tool, which means if there is a rough mask available for an image and we can somehow use some techniques to extend those masks to make it a better segmentation mask, then yeah, it can, supervised learning team would have more data and better data, I guess, um, to use their solutions and segment the images. And similarly, a rough mask along with a really good UI and some algorithms can also become a handy utility tool. The approaches or models we use for this particular solution pathway is point label aware superpixels, which is the PLASPIX, and the SAM and the SAM-HQ model. This is something we have been able to accomplish, except except the attaching the classifier part to the SAM and SAM-HQ the models. The another one was zero shot segmenter, where the idea was to take just an image and create a segmentation mask out of it with no prompting or no other manually entered data set. This eliminates the need to curate dense masks, and also it fine-tuned on some domain-specific data. It can, or it should have the ability to generate really good segmentation masks. The approach or models we use are fine-tuning SAM decoder and also using an auxiliary prompt decoder for SAM, which is something we or tweak from the AutoSAM paper. This is also something we have been able to accomplish. And then there's the interactive labeling tool, where we have done some of the base groundwork to actually build some tool like this, but we haven't gotten to the part of actually implementing the UI part of it, or a really good framework, I guess, to build the tool. But we will at the end give you a brief, I guess, walkthrough of how it could look like if we want to build it eventually. Um, this was the general roadmap when we started. After a couple of weeks, we wanted to experiment with a bunch of different techniques, generate benchmarks, compare results, see which one is better. As you can see here, the ones with the green tick mark are the ones we went ahead with. So we experimented with PLASPIX and SAM. We compared the results. SAM was better, and so we fine-tuned SAM decoder and got the final benchmark. The final benchmark is, I think, as I speak right now, we are awaiting the results. We were not able to train the patch classifier to build the entire inference wrapper, but technically it shouldn't be a very sophisticated problem. as far as we think. Similarly, zero shot segmenter is a very straightforward approach. We were able to do a POC. It is visible with point labels, and we were able to train the shot segmenter. So label propagator with point label aware superpixels approach. This is the PlusPix approach I was talking about. The approach is pretty simple. Um, it uses an algorithm to basically calculate superpixels or generate an initial set of superpixels. Superpixels are basically, you can say it's a group of pixels, or it's a parent pixel visually. And um, it uses an algorithm to optimize center locations in the feature space, and with some custom loss functions, which is distortion and lambda conflict. The distortion loss makes sure that the boundary of the superpixel generated um, is in agreement with the point labels that we have provided to the algorithm. And conflict loss makes sure that um, the conflicting regions corresponding to the point labels stay separated, I guess. So it generates these super pixels, and it obtains a single species superpixel region, and even it by iteration keeps converting the soft memberships to hard memberships, and eventually propagates the class labels within the parent superpixels to all the unlabeled regions of the image. And so at the end, what you get is something like this as an output. Oh, an important thing, this method uses a feature extractor, which extracts features from the image, and using that the features from the dense feature vectors from the feature extractor, and a couple different losses, it is able to calculate a super pixel membership for each class. Similarly, we're not trying to go a deep, very deep into the PlusPix approach and any tweaking we might have done. We haven't really done any tweaking, just experimented with it. But we found the SAM results much better for label propagation. And this is the approach, rough approach we used with SAM as a label propagator. We used the CV reef support data set with the full images and extracted patches in there. The way we extract patches from a full image is by using a dbScan algorithm to estimate the most densely packed regions of the image, or the most dense packed label points regions of the image. And we take these patches, we pass it through SAM. It happens in 3 runs because SAM can only do binary classification or segmentation, and so hence we pass it once for hard coral, once for soft corals, once for all the others. We blend the masks that we get from SAM, and we use we basically fine tune them with the point labels, and we get the final benchmark, which is the one I said we're awaiting results for. And these are the quantitative results we have from all the label propagation techniques, which is the PlusPix, and then 3 different solution options we have used for SAM and SAM-HQ, which is using SAM on full image with multiple prompting, using SAM-HQ on full image with multiple prompting, and then using SAM on high density patches with multiple prompting. As we can see, the best results we have for any of the label propagation techniques are with SAM on high density patches with multiple prompting. We get about 65.81% of MIOU, which is I think it's really great for an unsupervised learning technique. And after experimenting with so many different ones. Just to note here, the images used were resized to 1024 by 1024 in order to overcome GPU constraints for Plastics, and also to keep a standardized set for all different experimentation we have been doing. And this could also have played a role in lower performance. Um, but yeah, that sums up. And we have some qualitative results as well for everyone to look at. This is what we get from the superpixels approach and a full image, as we can see, since it kind of um, uses a lot, it generates some membership along with the knowledge of feature extractor and the point labels. It comes very close to generating the mask boundaries. This is the one with SAM-HQ with full image, and this is the one with SAM with high density patches. Um, Here, in some parts, we see some of them miss out, but overall, when we look at or compare the three visually, um, this approach gives us the best results. This might not be the best images for comparison visually, but yeah, I, I hope you get the gist of the approach. But yeah, that sums up the label propagation part. I believe we are taking questions at the end, Dorian, or should I pause right
Yeah,
now
no, I
if
think it's good
anyone?
to have some questions if there are any related to this.
Yeah, well, first of all, amazing results. I'm really impressed by what you were able to achieve in the 10 weeks that you've been giving. I was particularly looking forward to this part, the label propagator, as this would be something that could greatly help out with how we label our data sets and our efforts, and also using potentially this extracted masks as training data as well. What do you think your main takeaways from these 10 weeks was, and are you confident that with the 66%, which is very high for unsupervised, do you think that those masks can be potentially used for training a supervised model in a proper manner?
Um, for training the supervised models, that would be a tricky part to answer. I Especially because I'm guessing the accuracy of masks with human annotated dense masks, it's probably much higher. I'm not sure anyone ever tried to put a number to it, but I would guess it's above 95 or something. Just that makes something human annotated to use for supervised learning much better. But we do think there are some ways we can still improve the performance on these approaches by tweaking or combining it with some other algorithms. And and this is something we also talk at the end about, which is we talk about the future, some considerations or ideas we have on these all of the work we've done. And but I think the label propagation approach that we have experimented with, that we have developed, is definitely a really good option for the interactive labeling tool, or it can act as a really good uh, initial stab at segmentation, which then even a human can take in, and or some kind of, I guess, computer or image processing plus human method can take up to enhance the segmentation masks even more. Um, but yes, we as a team do feel fairly confident on the label propagation part, to answer your question.
Right, yeah, that's great to hear. I think we can proceed.
Cool. So uh, Ponia, I think, is on the call, who will take up the next part of the zero shot segmenter. Ponia, are you there? Or maybe not.
Hi, Masum.
Okay, perfect.
Yeah, thank you. Can you please continue sharing so that I can walk through the slides?
Definitely.
Yeah, can you please move to the next slide? Yes. Thank you, everyone, for the opportunity. So this is a continuation of the unsupervised learning sub-team. So here, Masoom talked about the label propagation approaches up until now. Now we would talk about a zero-shot segmenter, which is something that we aspire to do by looking at what the supervised team was doing, because they were working with dense mass and they were training deep learning models to come up with models that can do zero-shot segmentation on coral images. So we also wanted to do that, but with a slight twist. That is, we would not use the dense masks. Instead, we would use just the point labels. So just to give a comparison, if an image size of 1024 by 1024 is used, so with the supervised team, there is about 1 million pixels of information that is available to train or fine tune various models. But then with point labels, we would have in our test set or in our training set, we had around 100 point labels per image. So we are doing with just 100 points per image instead of 1 million points per image. So what we did is we took the same approaches that supervised team would take and do the fine tuning of the models. And instead, we did the fine tuning or we did the loss computation with just the 100 point labels. Uh, the three papers that we showed earlier in the literature survey, those were the three that we tried to implement. And we are showing only two because these are the two promising ones, even with dense mass. So we also experimented with these with point labels. The first one is fine-tuning SAM's mask decoder. So here we use a separate decoder for each class. So we want to identify hard corals and soft corals. So we fine-tuned two separate decoders, one for hard coral and one for soft coral. That is because SAM inherently is a binary segmentation model. The results that we get are not even close to what we obtained from label propagation, which is the mean IOU of 47%, which we tried on a single region. And then we tried something called as prompt overloader, which the supervised team would go in depth and explain what it is all about. So we added an additional prompt encoder, which generates a very rough mask for SAM to fine tune or to make it convert it into an accurate mask. And here again, we need to train two separate prompt encoders, one prompt encoder for hard corals and one prompt encoder for soft corals. And here we did it with just 100 point labels. The loss was computed during the training process with just 100 point labels, and the weights and biases were updated based on that. And on a single region, which is Seaview Atlantic Ocean, we found a mean IOU score of 58%, which is close to what we obtained from label propagation. But with label propagation, we also give the point labels as inputs. So from there, the information is spread out to the image. But here, this is a completely zero-shot segmentation. We only give the images, but we trained it on a training set with the 100 point labels per image. So it gives a comparable performance with the label propagator. So this gave us a very good confidence that point labels can also be used to train or to fine tune SAM. And also, you would see later in the presentation from supervised team that the fine-tuned SAM decoder performance is almost similar to using dense mass for training, whereas with the prompt overloader, that is 10 percentage points lower compared to the results from dense mass. Okay, so with this, we have an idea, which we are not yet implemented yet, but the idea is, in the data set, we only picked the reef support labeled samples. But then we have a big chunk of images, which is 8,680, which is from the original Seaview team. However, the interesting part is those images, they contain 100 or more point labels, that is between 100 to 300 point labels per image. So we have much more additional information compared to what we experimented now. So the idea is we could retrain the prompt overloader using the remaining Seaview data set. And we could get better results using that. Can you go on to the next slide, please? Yeah. So this is a sample qualitative result from fine-tuning of the mask decoder from one example. On the left is the output for hard coral. On the right is the output for soft corals. So you can see that there are some misses here. Can you go to the next slide, please? Yeah. So this is the best option that we have, which is the prompt overloader approach. Here there are two samples that we have highlighted. On the left is hard coral, on the right is soft coral. And the first one is the image, the second one is the ground truth, the third one is the predicted mask. So you can see that the predicted mask and the ground truth mask are pretty close to each other. And the IOU scores are around 58% here. Can you go to the next slide, please? Yeah. These are some of the key findings that we have from the unsupervised learning perspective. The first one is with the point label aware superpixels, that is the PLASPIX approach. What we notice is when we experimented with more number of point labels per image, the performance improves. To give you a comparison, the original paper, they used 300 point labels per image, but those images were much smaller than our images. They were 4 times smaller than our images. Okay, but then they had 3 times more data points to train. So the results that they got, which we showed in literature survey, those were by using really large number of point labels. But we experimented with almost 12 times lesser amount of information. So our results are lower compared to what the paper had reported. But that's expected. Okay. The second point is about SAM and SAM HQ. What we noticed is, surprisingly, by increasing the number of point labels, we did not see better performance from SAM or SAM HQ. What we noticed was the best performance was provided, was given by the models when we used around 50 to 100 point labels per image for the size of 1024 by 1024. So this is an interesting find for us. And, and based on these experiments, what we found is that SAM or SAM HQ is a very good utility for doing manual annotation rather than using the PLASPIX or the point label aware superpixels approach. And also with high density, that is by identifying patches that have high density of point labels, SAM performs much better. So with an interactive labeling tool, SAM can really be useful. That is SAM or SAM HQ can really be useful because the users can focus on the areas where there is an inaccuracy in the mask, and they could guide the model with point prompts, and they could get accurate segmentation masks. Could you go to the next slide, please? Yeah, the key findings that we have from zero-shot segmenter is that just 100 point labels are sufficient to fine-tune SAM. And the performance that we get is similar to label propagation, and it is close enough to a dense mask-based fine-tuning. So this is a very interesting find because this can be seen as the loss function being computed through a random sampling process, and that can also be seen as a mechanism for preventing the model from overfitting to the data. So it can be a generalizing way of generalizing the model for unseen images. So I think this is an important finding from what we experimented with zero-shot segmenter. Yeah, in terms of the future directions, what we want to do is we want to experiment with the high label density patches on point label aware superpixels approach as well, so that we can do a straight comparison between SAM and SAM HQ and PLASPIX to see which is better. Currently, we only did with SAM. The results we showed, which is the best result, that's from SAM. And the second point is, which I mentioned to you earlier as well, that is we could train an even powerful zero-shot segmenter by fine-tuning SAM using the CView point labels dataset that was unutilized because there are no dense masks available. But we don't have to wait for dense masks. We can use the point labels, and we could get dense masks. So what we could do is we could take the 8,680 images. We could use them as training set, fine-tune SAM, and then we could pass the reef support labeled images as input, get dense mass from this fine-tuned model and then compare with the hand annotated reef support dense mass and see how much overlap is available in the dense mass that this zero shot segmenter provides. So if the results are pretty good, then uh, we can stop doing hand annotation of dense mass, and rather we could use this as a first step, and then we could adjust the mass or we could fix some of the mass manually if needed. And with SAM, the mass generation can also be done in an iterative manner. So these dense mass can be fed as dense embeddings to SAM to generate a really fine-grained mask in an iterative manner, because SAM was also trained in a similar fashion. So I think these are the two future directions that we can explore from the unsupervised learning perspective, which I believe, or which I intuitively feel that would give good results for us to take it forward. So with this, we conclude the unsupervised learning team's work.
Thank you very much for the presentation, guys. Really nice. Looks very
Thank
good.
you.
We're also, although over half of the time yet already, so let's keep the questions to the end, and then we can head on to the supervised learning teams.
All right. Can everyone see the screen, the presentation here?
Yes.
Perfect. So hi, everyone. Thanks for the opportunity. I will be presenting the results from the supervised subgroup that focused on the YOLO V8 modeling approach. Here is the brief agenda for the presentation today. So we'll very quickly go through the challenge again, explain what YOLO V8 actually is, and then we'll spend some time on the data because it's very important. We'll have an overview and maybe explain some identified issues. We then go through some pre-processing stages and finally look into the modeling approach. That will lead us to the results and conclude with future work and our own conclusions and key findings. If you have any questions during the presentation, please interrupt. It's more interesting for everyone. And if you have a question, I think other people will have it. So please do it. All right. So very quickly, the challenge, as Dorian mentioned, is to bring down the cost of annotating underwater imagery. And a crucial application for that would be to estimate the benthic coverage of different coral groups and coral and benthic organisms as well. So this is one of the results of the YOLO predictions that we'll see later on during the presentation. But first thing first, the YOLO V8 model. So it stands for You Only Look Once, version 8, and it was originally a real-time object detection algorithm, as you can see here. And over the years, it evolved to do more things. And what we decided to use was instance segmentation. So we also have the localization information on top of segmentation mask. And yeah, you can also dive into the architecture, but we don't really have time today. I will skip these parts. Now the data. So let's start with the data overview, and we'll focus on the dense labels. Before we talked a lot about the sparse labels with like only point labels and so on. And what was used during the this supervised approach was the dense label. So what we mean by that is for each provided image in the data set, we get a dense mask associated with it. And on top of that, we also have individual mask for each coral instance. So in that case here, red would mean hard, I believe, hard coral, while blue would mean soft corals. And I only show like 3 individual masks here, but that's probably like 10, 15 for this image here. On the top left, you get like a random sample from the reef support dataset. So it's very like a broad range of different corals because they are taken across the world in 7 different regions and with different distributions of hard and soft corals, which makes the data set, when we look at the label distribution, very tricky. Hard corals are 5 times more present than the soft corals overall, and there is also variation within regions. In total, about 3,300 images were provided, but we'll see in the next slides that we were only able to use about half of them because of data issues. So let's now focus on the data issues we identified. The first table here is a summary of the provided dense labels per region and the number of dense labels that were usable. And for each region here, we expressed the reasons why we had to remove some of those images, basically. And now we'll basically describe all of these issues. The first identified issue was very low dense labels quality in one of the regions. So here you can see we get provided an image, probably a lot of hard corals on there, but like the segmentation mask is just red. So everything is hard corals, which actually makes it very tricky for YOLO to understand what's going on. Because what's YOLO expecting here is a bounding box with, like, for each hard coral instance with its segmentation mask. So we had to exclude this whole region, which I believe some of them are still good, but we decided to just remove this as a first step. The second issue we had identified was a bunch of empty masks. So just like probably some export issues that were fixed by reef support during the time of the challenge. So that was good as well. Another bit more sneaky issue that we identified was that there's a huge variation of image quality across different regions. So in Seaflower Bolivar, for instance, the average image from there is around 5 megabytes, while in Seaview, it's less than 1 megabyte. So that means that for a given image, there are like way more pixels coming from Seaflower than from Seaview, which could then lead to issues, as we'll explain, when we report metrics at the pixel level. Yeah, let's first talk about the mismatch labels that were also mentioned with the unsupervised work. So when the unsupervised team ran an EDA on that and an analysis, and they realized they compared basically the sparse labels with the dense labels. And every time there is a white cross here, it means there is a mismatch between the point labels and what's on the dense mask. So there are many images that have basically a lot of mismatch per image, and we decided to exclude them above a certain threshold as well. And at last but not least, an actually very critical data issue, I believe, is some data leak that we identified in the TD's Providencia. And the problem here is that for a given quadrat, we have overlapping images. So the actual issues is that, let's imagine that you have in your train split image 1 and image 2, when in your evaluation or validation splits and test splits, you have image 3 and image 4. And it's actually a very easy task for the model to segment image 3 if it has seen image 1 during training. So the problem with that is that it might overinflate the performance when you look at the overall performance across the whole data set. While in the wild, when you actually deploy it, it performs really badly, or like not the way you would expect it to perform. And we'll actually try to explain or evaluate the impact of this data leak later on the results that I will share. All right, so now that we have identified the data issues, I'll talk a little bit about the data pre-processing that we had to do to just make it trainable with YOLO V8. So it's actually a very simple process. And I will explain it briefly. So for each individual mask that are provided, we turn that into a line with numbers. The first number is the class number. So 0 would mean hard coral and 1 would mean soft coral. And then it's like X, Y coordinates of the polygon that makes up the contour of the segmentation mask. And it's normalized by the size, like the width and the height. So it's numbers between 0 and 1, basically. So we had to take care of this pre-processing step first before training. That leads us to the data modeling and data augmentation stages. When it comes to evaluation metrics, to be able to compare with other approaches, we decided to use the mean intersection over union and the dice coefficient. It's not the best metrics for instance segmentations because it loses all the information from the localization boxes, but it's a fair comparison with like other models that cannot do instance segmentation. So we decided to keep this to make it very easy to compare model performance. On the top right, you see like the train-val test splits methodology. I won't dive too much into this, but I would like to spend some time on the data augmentation we did for YOLO V8 training. So as you can see, we decided to use different techniques here. The first one would be to rotate, translate a little bit, play with the image contrast. But a key one was to actually, it's called the mosaic augmentation. We glue together, make a patch of images from the data set. So yeah, there are some good examples here. It's like basically 2 or 3 images that are glued together, and YOLO would see that. So it would actually, you could have like a lot of more, like many more combinations of mosaics with your data set. That actually helps with the generalization of the model. And without further ado, let's look at the results from this. The first thing to do was to establish a baseline quickly to be able to see how much improvement we could make. So we decided to pick those hyperparameters. So it's very basic. Basically, we didn't change much. It's a medium-sized model, training on 5 epochs on the full data set, and so on. So we could come back to those hyperparameters during the questions. And basically, we get already like very good results from the first training runs. So name IOU of 0.7 when we take into account the other, which is like the black pixels. And here you can see the training plots. So a lot of room for improvements. We could keep training. So that basically comfort us into the fact that we could still greatly improve the model, and we had to find the best hyperparameter combinations to squeeze the performance out of the model. Then you can see some of the prediction masks that the baseline was able to make. So on the left is the image from like a random image from the test set, the ground truth mask, and on the right is the prediction made by the baseline. So as you can see, it's able to find corals, contours actually, but it still makes a lot of mistakes with classification. So it doesn't really know if it's hard or soft corals in many cases. And this is the actual, at the pixel level, classification, we get this confusion matrix that summarizes the performance of this baseline, which is already quite good. So we decided to spend more time on finding the best hyperparameters, and we came up with those ones. So we increased the model size roughly. We trained for way more epochs, and we increased the image size and did some data augmentation. So first, I would like to show you the prediction mask from those best models. So as you can see, they are quite good, really, really better than the baseline. They don't make that many mistakes when it comes to classification, and they can more or less find all the coral instances in the image. And when we look at pixel level classification, it comes really close to perfect score. It still has some mistakes, like it makes mistakes between soft and hard, but it's very limited. And when we look at the training plots, we see that we reach kind of a plateau. We won't go any further. So that means that we've basically kind of squeezed out all the performance we could get from the data and those set of hyperparameters. And before jumping to my next slide, I would like to show you this, because we trained basically a model for each model size that was available in the YOLO V8 documentation. And the idea is that we don't really know the constraints or the hardware configuration that's available downstream. So we start from the biggest model sizes, which has among the best performance. And as we shrink the model size, we can see that we still retain a lot of performance. But those smaller models can actually run in real time on embedded device, probably underwater cameras and so on. So it's actually, it could be actually used in different contexts as well. And we provide all of those models as the outcome of the challenge as well. All right, so let's now try to break down those results because they are taken at the dataset, like the global level, and we'd like to look at the regional performances. If you remember, I mentioned like the data leak issue that we identified quite late actually in the challenge. And in this table here, what you can see is that the first line, so when the data is taken from all regions, we have the performance I showed you before, and then we actually evaluate on test sets for different regions. So the second line would be Seaflower Bolivar, and so on, Seaview Atlanta and Atlantic, and so on. And the last one is TD's Providencia. And it's not a surprise that it's the best performance because I think of the data leak. So we kind of identified the over reporting of that region for the model. We also summarize the weight that each region provides to the overall performance based on the number of pixels per test set, so per region. So you can see that, for instance, the overall score is made of 40% of the Seaflower Bolivar, but it contains only 25 images. But it's because images are so big in that data set and we report at the pixel level that it has a very huge impact on the overall score. So what we really need to do is to actually look at the region level to have an informed guess of like how the model will perform in the wild. Yes, that was it. Then two questions we wanted to answer was how well can a model generalize to ancient regions? And short story short, it can. So you can dive deeper into those summaries later on. But basically, we learned that it can generalize, but it's not as good, of course, as a model trained on the full data set. So there is some generalization power from a given region to another. There is knowledge transfer that's being done. And then the second question we wanted to answer was that, is it better to have like 7 different models, one trained per specific region, or a global model? What's the best performing configuration? So again, you can take a look at those summaries. We don't really have much time, but the short answer is that the global model is almost always better or equal to fine-tuned models on specific regions. Yes, except for one region, which is very small difference. That leads us to some future work that we could do. So one thing we could consider is that to expand the taxonomic scope, for example, including additional benthic organisms and functional groups to basically better understand the benthic coverage. We could also provide a way to continuously retrain the model when new data becomes available. And as the unsupervised team mentioned, we could also use the prediction from YOLO as a starting point for new regions or new data points. Another good one would be to establish a virtuous loop. So basically provide an interface for conservationists or researchers to upload their image. And it contributes to bigger data sets as they get like better results. And the final one, maybe finding ways to use small footprint models into, I don't know, underwater cameras to have like real-time feedback on the benthic coverage and things like that. So the key findings and conclusions, we validated the modeling approach that is YOLO V8. We think that it's highly suitable model for benthic segmentation. It has surprisingly exceptional performance, even on the most modest hardware configuration. And another takeaway is that one model is better than region-specific models. And last but not least, the dataset curation is very important and could yield much better overall performance, because in our case, we only trained on about half of the data sets because of the identified issues. That is basically it for the YOLO V8 subteam, and I will take your question now, or maybe we do it at the end. I have time.
Very nice, Arthur. I was impressed even also with that YOLO Nano has similar results actually than YOLO X, so it's very nice. In terms of time, I think it's best if we give Johan the opportunity to ask a question and then collect all the other questions in the end. So I don't know if you have any, Johan, for Arthur specifically.
Yeah, I already want to say thank you very much, Arthur. The results are amazing. Yeah, it was very nice, as Sako mentioned, also to see that smaller models do perform well, because that is potentially one of our ambitions as well, to look at embedded devices and if models can be trained to perform on those as well, particularly with videos format. I think we had a discussion, but what are your thoughts on that? Do you think that is a possibility with the results that you've currently attained as well?
I think it's definitely possible. When I looked at the model footprints for the nano size, one thing we would need to look at, though, is the regional performance of this model, because I haven't evaluated like the actual data leak issue on these models. So I don't really know like if the overall performance kind of flying or if it's in par with the best models with bigger sizes. But that seems promising.
That's great to hear. Well, I have quite a few questions, but I think it's best if we move on for now, and maybe I can also ask the questions individually at another time. I also have questions for the first team still, so we'll see how we can handle that after. But thank you, Arthur.
Cool. Yeah, for now, let's move on to the next team. And otherwise, you can also put them in Slack or plan on another separate short discussion on them. So thanks, Arthur. That brings us to the supervised SAM team, who can enlighten us with a nice presentation. Yep, we see your screen.
Okay, that's good. Yeah, so I would also try to be quick. So Here we explored different ways for fine-tuning SAM using a supervised fine-tuning approach and with further dense masks. So I would skip the literature survey as it is same as that from the unsupervised team. And so here is just the basic architecture of SAM. And then here we looked at one can, in addition to the input, also pass the inputs to the prompt encoder, which can be points, bounding boxes, or masks. And we actually used we also made some modifications here based on the different literature surveys or the different articles to see which approach would work the best for us. And the first one is the fine-tuned SAM decoder. So in this approach, we tried to duplicate the SAM mask decoder for each class and then fine-tune the mask decoder. And where we actually where we pass to it the inputs of each class of interest. So we pass the masks to the prompt encoder, and then which resulted in the dense embeddings, and then that was passed to the mask decoder. This was the first approach that we tried. And then the next approach is non-promptable multi-mask decoder. So here we create a model that can actually generate separate masks for multiple classes of interest. For us, it would be then two classes of interest, one for hard corals and the next one for soft corals. And then here, actually, it duplicates the learnable mask tokens and creates this the IOU token as many number of times as the number of classes. So here, the prompt encoder is discarded completely, and yeah, and there is a scope for fine tuning is limited over here because the number of trainable parameters are less. And when this was tried on the CView ATL dataset, the results were the lowest, I mean, in terms of the IOUs and so on. And so we also explored explored the approach of prompt overloader, which was also mentioned by the unsupervised team. Here, we actually leverage the SAM decoder's dense the dense embeddings input parameter. So what we do is we freeze everything, the image encoder, the prompt encoder, as well as the mask decoder. And we pass the input image through an auxiliary prompt encoder. And now this auxiliary prompt encoder, it actually produces a rough segmentation mask for the image. And what we use over here as the auxiliary prompt encoder is a harmonic dense net model with a CNN-based architecture. And this model is then used to generate the rough binary mask. And the HardNet 85, this is the model that we exactly used. And this has 47 million parameters. So it can also learn the fine-grained features as well. So, but the current architecture that we have that can only generate a binary mask. So which means that we need a separate auxiliary prompt overloader for each class of interest. So, and that's how we did it this time. So if I go to the results now, then over here in the results, we have the results for the fine tuning of SAM mask decoder, and where you can see that first we have the results for the CView Atlantic data set, and then we tried it with the reef support data set. Here we used the regions that were also used by Arthur to train YOLO V8. And then in the all data sets, we have all the reef support regions as well as the coral sec data. And after that, so, and then we have, if we look at the results from prompt overloader and the fine tuning of SAM mask decoder, then we can see that with the prompt overloader, we have much greater IOU as compared to the mask decoder. And that's in all the cases in with CVU Atlantic as well as reef support and all data sets as well. So with the reef support, we have actually the mean IOU is 67.50%. And then for all the data sets, we have it as 72.5%. And here are some qualitative results from the prompt overloader. So as you can see over here that on the left hand side, we have the hard corals, and on the right hand side, the soft corals. And then for 2 images, the ground truth mask, the predicted mask, and also the predicted blotches. And the future directions are that currently we just tried the prompt overloader with default parameters, but then we can also find the best hyperparameters for the prompt overloader. And yeah, and also we can also implement image augmentations, like the ones that also Arthur mentioned, and then also try some composite approaches. So if they would help us to get better results. So yeah, now I would hand it over to the YOLO V8 plus SAM team. Thank you.
Thank you. Um, just thank you, Iksa. I'll keep the questions for after Team West then as well. That's great. Yeah, so me and Cas worked on combining the YOLO V8 for which Arthur already presented with the SEM model. And the idea was to use bounding boxes as a prompt of the SEM model. So the concept is that we used the fine-tuned YOLO V8 model to produce a bounding box. As you can see in the image, it produces several bounding boxes around the hard and the soft coral. And then we use those bounding boxes as an input to the SEM to direct it towards the right places to create a segmentation mask. And then we combine all those segmentation masks into one segment, big mask. And we use the predicted class from the YOLO V8 to determine the class, because as I said before, SEM only outputs a binary mask, and hence we had to use the class from the YOLO V8. Interestingly, the performance of this combination is worse than it is for the separate models. So we expected a higher performance, but this was not the case. We did find that the SEM-HQ model performed slightly better, only 1 or 2 percentage points, but it is something. So the IOU is slightly higher for all classes, but still it's really poor, especially for class 1, which is hard coral. And then we kind of found a better performance for the FIT-H model, but yeah, that was not as clear as the difference between the SEM and the SEM-HQ model. So I would, if I have to choose it, the FIT-H is the best model, but yeah, that was by no means certain or significant. And so just for context, we did this with the SEM-HQ and the SEM model, just the untrained models, so vanilla models. And then we tried to combine the fine-tuned model, which was presented before, with the YOLO V8. So we hoped this would result in better performance, but we ran into some technical issues, because the bounding, to provide the bounding boxes as a prompt, we had to write a lot of code which was not in the scope of the project anymore. So what we tried was to use the bounding boxes from the YOLO V8, crop the image, and then provide it to the fine-tuned SEM. But yeah, this resulted in really low performance. So which is not really unexpected, because the cropped images are so different from the big images. So it was worth a try, but this definitely doesn't work. Yeah, and that brings us to the future direction as well, to try the fine-tuned SEM model with the YOLO, to combine the YOLO V8 model with the fine-tuned SEM model, but then provide the bounding boxes as a prompt rather than cropping the image. So
yeah, that was basically it. So the current architecture doesn't really seem to work, but there might be a possibility that with the bounding boxes as a prompt and fine-tuned SAM model, it might work better than the other solutions. So that was it for my part. I think that brings us to the Q&A.
Right. Well,
Thanks, Timo.
yeah. Thank you, Timo. Thank you, Isha, as well. Yeah, and I want to say nonetheless, even if the results were not what you expected, it's still great research and very valuable for us as well as a conclusion to well, looking at different research directions. It is surprising that well, it is interesting to see that the combination of the two models is not performing as expected. Same for I think right before, if I remember correctly, Isha, what you presented. The results seem very like 1% better, I think, in terms of IOU compared to the results from the first team using the points instead of the dense masks as for the training of the prompt encoder. So that is interesting to see as well. So yeah, thank you. I want to say a thank you to everyone who was involved in the research. Thank you to the presenters as well. There's a lot of information that was given to us in the last hour, so I have a lot to go over in the next few weeks as well before I can really give a conclusion as to what is going to be used or not for in our sense. But any research that was done is definitely something that we see a lot of value in. I think in terms of questions, yeah, like I mentioned, so many information that came in. So I think it's best if, for now, I review the slides again, review the materials that you came up with, and I'd really like it if maybe 1 or 2 persons per team would be willing to potentially share their email or have a short discussion, just because I'd like to explore every research has been explored individually and potentially have a short discussion on that. So that would be amazing if possible, if you're available for that. But again, I'm very much impressed as to what has been produced in the last 10 weeks, and I cannot express my gratitude enough. So again, thank you very much for your willingness to participate in this challenge. I hope you enjoyed it as well. I hope you got quite some things that you learned out of it as well. And of course, thank you to the Food Punch team, Sako, Leo, and Dorian, for helping us host this challenge, the second challenge. So yeah, I'm very grateful for all of this research.
No problem at all. It was a blast once more, Johan, and I learned a lot from all the people in the team as well, as I did last time. So super cool. Indeed, I think for the remainder of the questions, since we're well over time, if you reviewed the slides, just you can quickly post in the Slack for this meeting. If you can just post like a let us meet or something in there, I'm sure that some people from the team will be willing to give some more explanations in there. So that you can get to the nitty gritty, and we can also see what is actually implemented into Reefs report, which is also something to please keep us posted on. If you have the first go live based on that research, and we would all love to celebrate together with you. So you can post that in the Slack as well.
Right, that's 100% going to happen.
Very cool, very cool. So then we can see the fruits of their labor, I should say. Yeah, also from our side, guys, I love the process all over, and we use some really cool technology, also using the technologies that I at least was already familiar with in new and innovative ways. And I can't thank you enough for my own learning experience, but I obviously also hope that all of you also learned a lot throughout this project from your peers and from everybody in here. As I expect, Johan will be able to use this research to really increase well, the value that reef support can, but mainly the impact that they can make. And that's, of course, why we're all here in the end, to make a real impact using AI. So thank you all for your efforts over the last 10 weeks. It really warms my heart that you have been able to pour in so much of your free time to help the coral reefs of this world. And I expect this to be taken a long way by Johan and the team. So as a last time, within this context, at least for all of you, thanks, and I wish you a very, very fruitful day.